So today we're talking about enzymes. Enzymes are actually used to control or to maintain or regulate chemical reactions within organisms. And most enzymes are actually going to be proteins. So just like proteins, enzymes are made up of amino acids, which are made up of the elements carbon, hydrogen, nitrogen, oxygen, and phosphorus. Remember that there are 20 different amino acids. So because they're made up of proteins, the function of the enzyme is going to be due to, or the function is going to be a result of, how the amino acids interact or how they fold on each other. Thinking of the general enzyme reaction, we first have the enzyme by itself. The enzyme has a very specific shape at the active site. So this is our special shape. The enzyme is going to be very specific to a substrate. Substrate can also be known as a reactant. That's what's going to be worked on or changed. And then they will bind together in what's known as an enzyme-substrate complex. The substrate will be worked on while it's in the complex, and it'll be converted into proteins, which are eventually released. So here you have the enzyme sucrase, and it's working on sucrose, and eventually sucrose gets broken down into glucose and fructose. Now notice the names here, sucrase, okay? Sucrase, when you see that ace at the very end, that's how most enzymes are named. Ace implies that it's an enzyme, like catalase and polymerase as well. So enzymes are going to work to make a chemical reaction work faster. And because it's causing a chemical reaction or a metabolic reaction to occur faster, we actually call it a biological catalyst. So when we're talking about biological catalysts, there's a couple things that we want to pay attention to. Now, we're calling it biological because it's found in living organisms. Because it's a protein, it's made by the cells, so that kind of flows into each other. And one thing we need to keep in mind about enzymes is enzymes do display some unity and diversity. For unity, this means that the same organism or the same cells can produce the same type of enzymes, or even between different organisms we might have the same enzyme. Enzymes also display some diversity because within one organism we can have many different enzymes. Within one cell we might have more than one enzyme, and between different organisms we'll have different enzymes. Okay? They are specific, and we will talk about that in a little bit. But if you're a biological catalyst, this means that you speed up chemical reactions. And enzymes are able to do this by, or due to, their R group. So the R group of an enzyme is going to chemically interact, and I know this looks horrible, but it's going to interact or stress the bonds of the substrate. And remember that the substrate is the reactant of the reaction. It's what's being worked on. And so what this means is that it's actually going to make it easier to break or form bonds within that substrate. So between the substrates or within that substrate by itself. When we're talking about biological catalysts and talking about reactions, we really want to think about this activation energy. And this is the amount of energy it takes to get the reaction started. So it's like how much gas you need to put in a car to get it going, okay? Or how much fuel you have to put into the furnace to warm it up. And enzymes actually work by lowering the activation energy. So if you look in this first graph here, this says the energy released or the energy supplied. When you're looking here, this is the amount of energy, and if you look at the dotted lines, this is the amount of energy it normally takes for a reaction to occur. But if you have an enzyme, the amount of energy it takes for the reaction to occur is significantly less, so it's going to make that reaction happen faster and using less energy, so it's less expensive for the cell or the organism. And in the end, you end up producing the same products. Okay? Same product is made, but with less energy being used. So less energy used. So if you notice, you kind of have this backdrop of what it normally takes. That's our uncatalyzed reaction, our reaction that's not sped up. And then here we have our catalyzed reaction that is being sped up. And this is a lower activation energy compared to what we had over here. Okay? And so what we're saying is that in most cases, when we are actually going through with a chemical reaction, we want the enzymes to be here to speed that process up. There's lots of different reactions that occur in our body that occur very, very slowly. If you took a look at your book, we have a reaction caused by carbon dioxide and water forming together. So carbon dioxide and water would be two separate substrates that are being put together in an enzyme. And normally this is going to produce what's called carbonic acid, and that would be our product at the end. And this happens really, really slowly. Without an enzyme, your body will only make about 200 molecules in an hour, which seems like a lot, but it's actually very slow in terms of when we need it or how much we need. So with an enzyme being used to bring those molecules together, to put them together, then we end up actually making over 600,000 molecules within that same hour. And that's because the enzyme is going to lower that activation energy. Now when we're looking at these reactions, notice that here we have energy that is being released, and we also have energy that must be supplied. When energy is released out of a reaction, we're going to call this an exergonic reaction, and that's when it exits the reaction. So energy is released at the end of this process. But when energy must be supplied, we call this an endergonic reaction. And enter. Energy must be entered for it to begin. In an exergonic reaction, this is usually when we break down a molecule. So you have one big molecule being broken down, just like over here. In an endergonic, excuse me, you have a synthesis reaction occurring. You're putting something together. And so with a breakdown, this can be called catabolism. Or you can also think of a digestion. Okay? So basically what happens is our enzymes bring together these substrates, making it easier for these bonds to be either broken or to be formed. And it's going to overall increase the probability of the reaction to occur. Okay? That's the basic result of having those enzymes there. Now, today we are going to look at one of the other properties of enzymes, which is having a very specific shape. And remember that the specific shape of an enzyme is from the folding of the protein. So here you have the enzyme. You have its specific shape, which is known as the active site. The active site is going to be our location of where the substrate enters the enzyme. And again, the shape is very important. Okay? The shape of the active site is going to be specific to that of the substrate. So the substrate is our starting molecule or our reactant, and it is specifically shaped, or our enzyme is specifically shaped to our substrate. So this is actually sometimes called the lock and key. So there's one key per lock, so very specific. But now we also know more, we've learned more about enzymes, and we actually realize that there's an induced fit model. And what this means is that the substrate fits into the active site, and then we have a conformational change. Conformational just means a shape change. And what this really means is that our enzyme is going to fold in or hug or scoop closer to, fold in closer to the substrate. And one reason why this happens is because the substrate is attracted to the active site of the enzyme. And this is, again, due to the polarity and that hydrophobic or philic characteristic or tendency, okay? And so here you have that folding in, that conformational change can be seen right here. So again, we want to make sure that we take a look or review what is special about enzymes, what is the relationship between the enzyme, substrate, and products. You want to look back over that diagram of the entire process. And you also want to be able to describe the difference between the anabolic reaction and catabolic reaction. And just as a little hint or reminder, anabolic is a synthesis reaction, and catabolic is a breakdown reaction. Okay?